Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. Assalamu alaikum everyone. Welcome to World by Zoology. This is Iram Zafar. The topic which we are going to discuss today is Fasta format. So we will discuss in this video tutorial about what is Fasta format, how can you retrieve Fasta format using Ncbi database. So first of all, let's have an overview of Fasta format. So Fasta format in bioinformatics or biochemistry is a text-based format for representing either nucleotide sequence or the sequence of proteins in which the nucleotides or amino acids are represented using single letter codes. For nucleotides, for example, A is for adenine, C is for cytosine, G is for guanine, T is for thymine, and U for uracil in messenger RNA. So for amino acid, for glycine, it is represented by G. So this is the FASTA format using single letter codes. So first of all, the FASTA format originated from the FASTA for software packages, but it has now become universal standard in the form of bioinformatics. So for the processing of software like Ncbi Blast for primer designing, they require the FASTA format to be processed. So the simplicity of FASTA formats make it easy to manipulate and parse sequences using text processing tools and scripting languages like the R programming language, Python, Ruby and Perl. So let's go to our screen to see how can you retrieve FASTA format using Ncbi. So first of all, open your Google Chrome and in the search bar write Ncbi. So the first search will be the National Center for Biotechnology Information. Click over it. This is the home page of Ncbi. By default, here it is written all databases. So you have to retrieve the nucleotide sequence in the FASTA format. So you have to select the nucleotide using the pull down menu. So select nucleotide over here, and here in the search bar you have to write the name of the gene or the protein of which you want to retrieve the FASTA format of the nucleotide sequence. So let's suppose I want to retrieve the nucleotide sequence of NRAS of Homo sapien. So I will write NRAS and click search. So the first search will be NRAS proto-oncogene GT-phase. It is the summary of the gene. So I have to open the RefSec. Here it is written RefSec transcript, RefSec protein, RefSec gene. So I want to retrieve the nucleotide sequence of the whole gene. So I would like to open it RefSec gene. So after pressing RefSec gene, you will have the summary in the GenBank. So you can retrieve also the FASTA sequence using GenBank page. Here it is written FASTA or graphics. So here it is also written Homo sapien NRAS proto-oncogene GT base protein है. It is GT base protein RefSec gene, the number of specific number in the genomic project on chromosome number 1. So here it is written FASTA. If you click over the FASTA, you will have the all the sequence of the gene in the FASTA format. So the description line. So the FASTA format always started with the greater than symbol. So it is the description line or the header line or the identifier line which begins with the greater than symbol, and it is started here. It is also written the accession number and an additional information about the gene. So here it is also written Homo sapien, means it is present in the Homo sapien. This is the sequence of the NRAS gene present in the Homo sapien. NRAS proto-oncogene means it is a cancer causing gene which converts into cancer causing gene if there is mutation. So it is also a GT-phase dependent or GTP dependent protein. So RefSec gene, the specific number of that gene on it is present on chromosome number 1. So it is the original sequence or the one letter code for the amine for the nucleotides present in the NRAS gene. So it is the whole sequence of NRAS gene present on the chromosome number 1. So this is the faster format of the NRAS protonco gene. So always remember that this sequence always starts with greater than symbol, and if you want to put this sequence in any of the software or tool like run blast or pick primers, so they require the faster format of the nucleotide sequence. So you have to select the whole sequence starting from the greater than symbol, then you have to copy and paste it in the run blast or pick primer. So this is the faster format, or this is the format you can retrieve from Ncbi. So you can also send the file by using this send to option by complete record coding sequence gene feature, or choose destination. You can also choose the destination, like you want a file for it, so I want a file for it, or I only want the coding sequence of this faster format, or I would like to choose here the faster nucleotide sequence because this is the nucleotide sequence, not the protein sequence. So click over create file. So this will begin to download over here. So after complete download, open it. This will open it in the notepad sequence. So this is the sequence of your whole FASTA format using the Ncbi. So I hope you understand this lecture. Allah Hafiz. Please subscribe, share and like my channel.